All right, so we're going to take a look at Chapter 8b, looking at the first step of photosynthesis, the light-dependent reactions. And in this video, we are going to be looking at this you must know here, how photosystems convert solar energy to chemical energy, how linear electron flow in the light reactions results in the formation of ATP, NADPH, and O2, oxygen, and how the formation of a proton gradient in the light-dependent reactions is used to form ATP from ADP and inorganic phosphate by ATP synthase. All right, so let's talk about the reaction of photosynthesis. And before we do, let's zoom out a second and take a look at where we're at. So remember, we're in the leaves of plants, and we're looking closely here at a plant cell, the organelle, the chloroplast, and inside this organelle, we have our thylakoids, these stacks, these individual pancakes, and these grana, these stacks of thylakoids. And let's just take a little overview look at what is going on in this chemical reaction. So photosynthesis, photo means light, synthesis means to make. So we're going to be making something using the energy of light. So here's our solar energy from the sun, enters through the leaves and makes its way all the way here to the surface of the thylakoid. We have some water coming in, some carbon dioxide coming in, and we have some oxygen coming out and some C6H12O6, glucose, or it doesn't have to be glucose. Photosynthesis also makes other carbon, hydrogen, oxygen atom or molecules, and they follow this general structure. So every one carbon, there's two hydrogens and one oxygen. So that is what gets produced in this reaction. And in this reaction, you might notice that there are kind of two sides. So there's this side on the left and this side on the right. We're looking at two types of reactions. We call this one the light-dependent reactions because it's dependent on light for energy. On the right-hand side, we call it the light-independent reactions. It happens independent of light. And we also sometimes call it the Calvin cycle. All right, so here is a closer look. So if we were to go inside this chloroplast, here we have, again, here's our double membrane, here's our thylakoid and our grana, and we're looking at, on the left, our light reactions, our light-dependent reactions, and our light-independent reactions, or the Calvin cycle. And notice that, let's just focus on the left-hand side for now. Notice that light and water are coming in, they're hitting the thylakoid, and oxygen is getting produced. Notice that there's also some energy-carrying molecules here. These might be familiar to you. So we have ATP and NADPH coming out of this reaction. So we're going to be making some of these high-energy molecules. And these low-energy molecules, NADP plus and ADP, this is our oxidized version of NADPH. It's lost its electrons, it's lost its hydrogens. And here's our lower energy form of ATP. We've lost the inorganic phosphate. So now this is going to get recharged by this reaction, and then it will be like a cycle. We'll come back to this side in a second, but in this video, we're going to focus on the light reactions. All right, so just a quick overview. The light reactions, or the light-dependent reactions, the whole point of this is to take solar energy, energy from the sun, and convert it into a new form of energy, chemical energy, energy stored in the bonds of molecules. So overall, we're going to be starting with energy in the sun, and the energy is going to end up at ATP. ATP is the usable form of energy for our cells, but energy is going to be shuttled around in different ways. We know that energy can be held by energy-carrying molecules, the electrons in NADPH, or ATP. Energy can be stored in sugar molecules, right, in the bonds of sugar molecules, but eventually, after cellular respiration, all that energy will be placed in ATP, which can be used to power anything in the cell. And then the second step we call the Calvin cycle. The Calvin cycle, like I kind of showed you before, uses chemical energy. So we're going to take these high energy carrying molecules, and we're going to use this energy, kind of like, think of these as like your batteries, to power the reaction of basically reducing carbon dioxide. Remember, reducing means to gain hydrogens or electrons, oil rig. Reduction or oxidation is loss, reduction is gain. And so we are going to be gaining hydrogens and turning this into a high energy molecule. Now that molecule down here has a bunch of hydrogens and electrons, and that can be broken up in cellular respiration to make ATP. All right, so let's take a look at like the broad picture, and this picture might stress you out a little bit, but we're going to go through step by step. So I just kind of want to show you what's going on generally, and then we'll break it down. Okay, so if this is overwhelming, that's all right. Just take it in for a moment. Try to notice what words you recognize, what structures you recognize. And let's just start with, where is this picture? What are we looking at here? So you might notice that we have a phospholipid bilayer here. Now, this is not a cell, but this is the thylakoid membrane. So remember how we looked at in our previous, where did it go? We can look here. Each of these stacks is a thylakoid, and it's made up of membrane. So we're literally just looking inside one of these pancake stacks. If you were to open it up and look inside, this is the membrane of one of these stacks. So this is one pancake, right? And you have your membrane. And embedded, you have these items called photosystems. These are a network of proteins and also pigment molecules that are going to be working together. And then outside of the thylakoid, you have the stroma. The stroma is the fluid that's surrounding the inside of the chloroplast. And inside, you have the thylakoid space. So this is the inside of the thylakoid. It's hollow, it's just space, and things can happen inside here. Now, overall, I want you to notice again how we have light coming in here and we're generating some ATP. So this right here, we're looking at the light-dependent reactions. This is the light-dependent reactions. It happens in the thylakoid membranes. And eventually, you're going to be taking your light, okay? You're going to be making some ATP. And notice the other thing you're going to make is NADPH. So NADPH has been reduced. It's gained that extra electron and that hydrogen. And now this molecule has energy that can be used later to power the Calvin cycle. So really the whole point of the light-dependent reactions is to convert solar energy into chemical energy in the form of NADPH and ATP, right? So these molecules now are holding the energy that was originally in the sun, and that's going to be like the battery packs to power the Kelvin cycle, which we'll look at next time. Okay. Now I want you also to notice, we'll get more into this in a moment, but notice we have some enzymes here. So NADP plus reductase, ACE is an enzyme, so we'll look at that closely. ATP synthase, remember we know about ATP synthase from cellular respiration. We see here that if we have a high concentration, this little bar here means concentration, a high concentration of hydrogen ions inside the thylakoid space and a low concentration of hydrogen ions outside, we have a concentration gradient. We have a chemiosmotic concentration gradient or electrochemical gradient. Basically, we have a high concentration of hydrogen ions, that's a chemical gradient. We have a overall positive charge compared to the more negative charge down here. That's an electrical gradient. And we can also call it an osmotic gradient because osmosis is a diffusion of molecules from high to low concentration. So these chemicals are going to be diffusing from a high concentration here to a low concentration. So an electrochemical gradient or a chemiosmotic gradient is what we're developing inside the thylakoid space so that those hydrogen ions can diffuse down using ATP synthase. They're going to diffuse into ATP synthase, come out into the stroma, and that is going to turn our ATP synthase and help phosphorylate ADP to ATP. So we are going to add on that last phosphate group that requires energy to put on that last phosphate group. And now we have a high energy molecule, an endergonic reaction here, and we are now going to be able to use that energy in the Calvin cycle. So let's break it down piece by piece, and let's just start right here. So what's happening when light is hitting this thing called the photosystem? So let's look carefully. So we're in our light-dependent reactions, right? We are in the thylakoid membrane, and we have this purple piece here called a photosystem. So a photosystem is a mix of proteins and these chlorophyll molecules here. These are pigment molecules that have the special ability to absorb sunlight. So light travels in waves, but we can also talk about light as traveling in photons. Basically, it's like packets of light that are hitting our photosystem here. And when the molecule here, this is a chlorophyll molecule, when the chlorophyll molecule, this pigment, receives a photon, it's going to become excited. So there are actually electrons in this pigment molecule that are going to basically absorb that energy from the sun. It's going to absorb that photon and become a high energy electron. If I can show you a picture here, if this chlorophyll molecule, so these little circles here are representing individual chlorophyll molecules, if the chlorophyll molecule absorbs a photon, it basically excites something called an electron. There's an electron in this chlorophyll molecule that becomes excited, or another way you can think of that is it now has gained energy. So the electron has gained a bunch of energy, and it got that from the sun. It absorbed that photon, that piece of the sun, and it's excited. All right, so it's at a higher energy state. Now, what happens is this electron is going to be passed to each chlorophyll molecule. So it's going to be passed from here to here to here, kind of like playing hot potato, right? You're passing this electron along until eventually it's going to be accepted by the primary electron acceptor. So there's going to be this spot here that accepts that electron, okay? So overall, the first step, we are absorbing a photon, exciting an electron, and passing it around until it eventually gets to our primary electron acceptor, okay? Now, if you were to look at it up close, what are we really looking at? The purple piece, it's just proteins, right? These are our alpha helices and beta pleated sheets, our secondary structure. And we can see close up our chlorophyll is this green parts. These are just molecules, right? They're pigment molecules that are absorbing light. So this is really what it looks like, but here's like an artistic depiction. All right, so we just talked about this idea of a photosystem, but let's take a closer look at what a photosystem is. So a photosystem is a collection of chlorophyll molecules. So all of these green pieces here are individual chlorophyll molecules. And there are two photosystems that are important in the thylakoid membrane. And they act as a light gathering antenna complex. Antenna means like they're like searching for the sunlight and they're grabbing onto the energy from the sunlight. The first one is called photosystem 2. Okay, now that might kind of sound funny. Why would we call the first photosystem 2? The reason is it's because it was discovered second. And inside photosystem 2, we have chlorophyll A molecules, which absorb a certain wavelength of light, so 680 nanometers. And if you were to look at the electromagnetic spectrum, the visible light spectrum part, we would be looking at red light. So if we go back to our spectrum, 680 nanometers around here, so that's like red light. So this special photosystem absorbs red light. All right, so next, we have something called the photosystem 1, which has chlorophyll B. Chlorophyll B is very, very close to chlorophyll A. It's just a couple atoms that are different, right? From our last video, we learned about that. And this one absorbs slightly different wavelengths, but still red light, just a little bit longer wavelengths here. And just like we saw before, here's just another picture. A photosystem is a collection of individual chlorophyll molecules. A photon will hit it, excite an electron, and pass it around until it gets to its primary electron acceptor, which will now have those electrons. So remember, we're talking about moving electrons, which is another way of talking about moving energy. We're transferring energy or electrons. All right, now just like we talked, there's something called linear electron flow. Basically, that means electrons are moving in a line or they're jumping around from place to place. So you can kind of trace their movement, kind of like moving in a line. And eventually, those electrons are going to go to produce ATP and NADPH. That energy is going to be held in the bonds of ATP and NADPH, which can be used later. Now, we can track the movement of electrons in the light-dependent reactions as a series of steps. So we're going to take a look at each step. How are electrons moving from this photosystem 2 here all the way to our NADPH and all the way to our ATP. So let's track the energy. We know the energy starts in the light. We just talked about how the energy is going to be passed around. These electrons are going to be passed around here. How is it going to be moving eventually to get into these molecules here? So let's take a look at each step. Okay, so let's take a look at this picture. Where are we at? So we are at photosystem 2. So if we were to look back at this big picture, we are looking at photosystem 2. Remember, even though it's the first photosystem, we call it 2 because it was discovered after photosystem 1 by scientists. So we're here. We're looking at this, and we're just going to look at it kind of a different picture. All right, so notice here the first step. A photon, so here's our photon, is going to hit a pigment in the light harvesting complex of photosystem 2. So the light harvesting complex means all those pigment molecules and proteins together like we saw before. And its energy is passed among pigment molecules until it reaches P680. So this is a special chlorophyll molecule. Next, it's going to excite an electron. So it's going to pass energy or electron to this chlorophyll molecule here. And it's basically going to increase the energy of that electron so much, and eventually it's going to be accepted by this molecule called the primary electron acceptor. So our first excited electron, remember, means now it has a bunch of energy. So the electrons were excited by the sunlight, and it's eventually going to be accepted by our primary electron acceptor. You can also call it P680 plus, but really I just refer to it as the primary electron acceptor. Now, this chlorophyll molecule right here, it's passing its electrons to the primary electron acceptor. So now it's lost electrons. So in order to replenish the electrons that were lost in this chlorophyll molecule, what we're going to do is we're going to take water, okay, H2O, and we are going to cut it up. We're going to split it. So when you split water, you're going to pull off these hydrogens. So now you have two separate hydrogen ions. And remember, hydrogen comes with an electron. But if you just have H plus, that's just a proton by itself. So where did the electron go? The electron goes here. So notice there are two electrons for each of the two hydrogen ions, and these electrons are now going to replace the electrons that left P680 when they were passed here. Okay, now in the process, now you have these hydrogen ions floating around. You're also having some oxygen left over. It has half O2 because you have basically an oxygen molecule or oxygen atom, but oxygen doesn't travel by itself. It doesn't ever travel just as O. So it's going to find another oxygen and bond to O2. But it's just showing you here that originally, right as these hydrogens are pulled off, you're going to have an oxygen atom by itself, so half of O2, but that oxygen atom is going to find another one and bind to it to make O2 eventually. All right, so the oxygen atoms, like we saw here, combine to form O2. So multiple oxygen atoms that are hanging out will find each other, form O2. Now you have these two hydrogen ions released into the thylakoid space. And so now if you look at this picture here, here's kind of what we saw before, but kind of with everything else. So you have your light hitting, you have the energy pass, the electrons pass to the primary electron acceptor. Oop, we got to replenish these electrons. So we're going to split water. And when you split water, you're going to have two hydrogen ions hanging out by themselves. Where did their electrons go? Their electrons went here to P680. And then you have those oxygen molecules that are going to be created. And this is why you're able to breathe, because in this process, plants are producing oxygen, right? And this oxygen will leave the plant and you will breathe it in. But what happens to these hydrogen ions? Well, now we're building up a high concentration of hydrogen ions in the thylakoid space. So eventually they're going to go down to ATP synthase, down their concentration gradient, and help to create ATP later. So that's kind of what the whole purpose was here. So we've created a higher concentration of hydrogen ions, but where do we leave off? What happened to that electron that was passed to the primary electron acceptor? What is its next fate? So let's take a look here. So we've got to the primary electron acceptor. What happens next? Well, these electrons are going to be passed down an electron transport chain from photosystem 2 to photosystem 1. And it's going to be releasing energy, and it's going to form a proton gradient across the thylakoid membrane. So kind of like we saw here, our goal is to try to make even more protons and place them in the thylakoid membrane. So let's look closely at what's happening here. So we have our electrons, and these electrons are going to be pulled towards this new molecule here. This is a protein, and this protein is pulling on the electrons. It's more electronegative, so it's kind of pulling the electrons towards its direction. And as the electrons are being pulled in this direction, they're going to next be passed to this called the cytochrome complex. And in this process, we'll take a look next. Eventually, ATP can be made here. Then we're going to pull the electrons over here to this protein until eventually the electrons are going to be moved to this chlorophyll molecule, P700, and this is chlorophyll B. And these electrons now can be passed to the primary electron acceptor of this photosystem 1. Now these electrons need to be replaced, and so light is going to hit the chlorophyll molecules here again. Electrons will be passed until they reach P700 to replace the electrons that were lost. Okay, so light is supplying the energy to keep this P700 full of electrons, because when it loses electrons, right, so it's going to accept and then lose it here, we need more electrons to be replenished. And so light will provide the energy. It will be converted into electrons, which will now replace the electrons here. Now, if we look at the picture here, we can kind of trace the electrons too through this yellow line. So notice electrons are being passed to this protein molecule. And remember from cellular respiration how electrons in an electron transport chain can supply energy to pump protons against their concentration gradient. So there's a lower concentration of hydrogen in the stroma on the outside here. And so the energy from these electrons are going to pump the hydrogen into the thylakoid space to even further increase the concentration of hydrogen ions so that we can use our ATP synthase later. So the purpose of the electron transport chain is to pass the electrons eventually to NADPH, but also to use some of the energy from the electrons to power the pumping in of the hydrogen. Now, what happens here though is the electrons are going to lose their energy along the way. As the electrons are being pulled, some of their energy is being used up here and being used up here. So that's why in photosystem 1, we basically need to re-excite and re-energize the electrons. So we're going to take in some more light here, and we're going to re-energize the electrons, basically so they have more energy so they can go and charge up NADPH. All right, so that's where we left off here. So our electrons are being passed, we're making some ATP. Next, if we take a look at where we left off here, so next the electrons are going to continue to be passed until they reach their final electron acceptor, the last molecule that will be accepting the electrons, which is our enzyme called NADP plus reductase. So let's kind of skip over here. I skipped this one right here. Okay, so I explained all the other slides. Let's see, we talked about that. Okay, now NADP reductase, this enzyme will catalyze the transfer electrons and H plus and to NADP plus. So basically, in order for us to attach this H plus to the NADP, we're trying to reduce this molecule, we need some electrons, right? The hydrogen, the H plus, and the positive NADP plus, those are like charges, they're going to repel. So these electrons are going to be added to this hydrogen and this NADP plus. So now you have neutral. So now you have NADPH, which is neutral, because these two negative charges and these two positive charges will now just become a neutral NADPH. But now this NADP plus hydrogen plus electrons, now these electrons are in this molecule. Now this is a high energy molecule, and we can use that for the next step later, the Calvin cycle, to power the Calvin cycle. All right, let's see if I missed anything here. Just going back for a second. Okay, so we got this. Yes. Okay, so I went through all this. If you want to go back to the slides and kind of look through each step here, you can go back and look at each step by step as I explain it. All right. Now I want to briefly show you and remind you that we had looked at an electron transport chain in cellular respiration. Remember, in cellular respiration, we were in the mitochondria, and we generated a high concentration of hydrogen ions in the intermembrane space. So the space between the inner and the outer membrane, this kind of darker space here, we had a high concentration of hydrogen ions. And we were using ATP synthase to diffuse these hydrogen ions into the mitochondrial matrix, this lighter space here, to make ATP, right? And we got these hydrogen ions into the intermembrane space by using the electron transport chain. So the purpose of that electron transport chain is to use the energy of electrons to shuttle hydrogen into the intermembrane space. So that was in the mitochondria. In the chloroplast, where are we? So we are increasing the hydrogen ion concentration inside the thylakoid space, and we are pumping out those hydrogen ions into the stroma, so this fluid part here. So it kind of is like opposite of the mitochondria here. All right, so notice here that we are making some ATP as we are diffusing hydrogen ions down their concentration gradient. So electron transport chains are really important. Their job is to generate a concentration gradient, a chemiosmotic gradient, right, or an electrochemical gradient, where now you can use ATP synthase, the energy from passive diffusion here, to generate ATP. All right. So just to recap, where are we? What are we doing? We are in a leaf. We're inside a leaf cell. We're inside a chloroplast, and we are in the thylakoid membrane of one of these pancake stacks here. That's where the electron transport chain and the light-dependent reactions are happening in photosynthesis. Now, here's a little model that we can kind of use to understand how energy is moving around in the light-dependent reactions. And I remember when I first learned this when I took AP Bio, this picture was quite confusing to me. But I'm going to kind of explain it to you and see if you can understand kind of what's going on. Basically, this picture is trying to show you how energy is being moved around and how electrons are being passed. So, where does all the energy come from to make all these electrons move around? Well, initially, it comes from the energy of our photon, our packet of light that is hitting our chloroplast molecules. And basically, that photon excites our electron or gives it energy. So you can kind of compare it to this mechanical energy here of someone hitting this right here. And now we are increasing, we're boosting the energy of the electron, right? So our photosystem 2, we are boosting the energy of the electron and we're passing it to the primary electron acceptor, this guy right here. So the electrons will be passed to different chlorophyll molecules until they get to the primary electron acceptor. Then this electron is going to be losing some energy. We're going to be making ATP. And if you think about, okay, where are the electrons making ATP over here? Well, we can look at this picture. The electrons are being used. Now they're losing energy to pump the hydrogen in to help make this ATP. So we're going to sacrifice some energy in the process to make a little bit of ATP. We need that ATP so that we can go power up other things. So we're kind of like charging up some batteries to power the next reaction. So if you look here in this picture, it's kind of comparing that to like turning this and making ATP. We're losing some energy in that process. And so now you've lost some energy. You've used that energy to shuttle hydrogen into the thylakoid space. So let's get another photon of light. So more light will hit photosystem 1 and it will recharge that electron. And that electron will be accepted by the primary electron acceptor of photosystem 1. And then this electron will eventually be passed to NADP+, and NADP reductase will add on that hydrogen and those electrons to NADP to make NADPH. And now we have our energy carrying molecule. So we can take our NADPH and our ATP to the Calvin cycle to power those reactions. All right, so once again, the light reactions, we talked about this and how they're compared to the electron transport chain in cellular respiration. If you want to pause this slide and look closely at it, you can, but I've already explained what's going on here. Okay, so overall, in photosynthesis, sunlight is supplying the energy to move electrons around and shuttle energy around. So sunlight is going to power the pumping in of hydrogen ions into the thylakoid space, right? Because it's exciting those electrons, it's pumping those protons, it's forming the gradient, the hydrogen gradient inside the thylakoid space, and it's basically supplying all the energy to make ATP, and it's helping to attach that last phosphate group to ADP to make ATP. Now, sunlight is supplying the energy in photosynthesis. But when we were talking about cellular respiration, where did the energy come from to build NADPH or NADH and ATP? The energy came from oxidizing glucose, from pulling off the electrons and the hydrogens of glucose and using those electrons to generate ATP and NADH, right? Now, that's the main difference between the two. Sunlight's providing the energy to move electrons around. In photosynthesis, respiration, you're using the energy of the electrons that are already in sugar. So we're thankful to photosynthesis because without this process, we wouldn't even be able to make glucose in the first place. So all the energy that's found in this molecule was originally in the sunlight molecule, which now passed its energy and converted its energy to chemical energy by moving these electrons around in the light-dependent reactions. All right, we talked about this already. Here's the electron transport chain in the mitochondria. The same thing happens, or a very similar thing happens in cellular respiration, but the only difference is that we're getting the energy, right? We're getting the electrons for NADH from our glucose molecule, right? Those hydrogens came from oxidizing, pulling off the hydrogens of glucose. Those electrons came from that glucose molecule to generate ATP eventually here and to push these hydrogens against their concentration gradient so that they can generate ATP here, right? Now, in our electron transport chain in photosynthesis, the electrons come from, let's see, I have a picture here. The electrons come from the sun, right? The sun excites the electrons and it allows this whole process to happen. So that's a key difference. Another key difference is the electron carrier in photosynthesis is NADPH, whereas in cellular respiration, we're using the electron carrier NADH. Okay, so cellular respiration, we're using food molecules to generate ATP, right? We're transferring chemical energy. This chemical energy came from food, from the hydrogen and the electrons in food. In photosynthesis, we get our light energy is being transformed into chemical energy to make ATP and NADPH. All right, so here's kind of another summary slide. I've already gone over this, but this basically is all of the light reactions. So just to go through it one more time, if you want to skip through this, you can, but just kind of to get it in your head, let's take a look at all the steps again. So we have light striking a chlorophyll molecule in photosystem 2 in the thylakoid membrane of the thylakoids here. And we are passing electrons around until they get to our primary electron acceptor here. Now we need to replenish those electrons, so we're going to split water, pass the 2 electrons that hydrogen originally had here, and we are going to now have hydrogen ions. Those ions have lost electrons, so they're just positively charged. And you're going to have some oxygen be made here. Those electrons are going to be passed along the electron transport chain, and the purpose of this electron transport chain is to pump hydrogen ions into the thylakoid space, which will eventually go down its chemiosmotic gradient and synthesize ATP. Now, those electrons have lost their energy by pumping those hydrogen ions, so they're going to get recharged by another photon of light, and they are going to be passed eventually to our primary acceptor in photosystem 1, which will go down another electron transport chain to eventually be accepted by our final electron acceptor, NADP+, reductase, which is an enzyme that will catalyze the reaction to add on these hydrogen ions back to NADP+, using the electrons that we had passed around here that originally were from the sun. And now you have your high energy molecule in your ATP, charged batteries, ready for the next step, the Calvin cycle. So if you look here, let's see if I have a better picture actually. Okay, here. So in summary, you can kind of see like the basics. We've now taken light and some water. We split the water. We've made oxygen, and we've taken the electrons from the light that the light has excited, and we've passed them to these high energy molecules, which will now power the second part, the Calvin cycle. And we'll take a look at what happens here in the next video. See if there's anything else. Okay. I think we talked about that. So if you want to look more closely at basically the summary of the light-dependent reactions, you can look at this slide here and pause it. All right, so just to summarize, like a little quiz for yourself, where did the energy for photosynthesis come from? From the sun. Where did the water come from? Came from the roots or even from the stomata, water vapor coming in through the leaves. Where did the electrons come from? The electrons came from water, right? From the hydrogen ions in water. The hydrogen atoms, when water is split, the electrons are going to be accepted by the chlorophyll molecules. Where did the oxygen come from? The oxygen came from splitting water. Where did the hydrogen ions came from? The hydrogen ions came from splitting water, cutting off these hydrogens. Where did the ATP come from? The ATP came from our chemiosmotic gradient, from the high concentration of hydrogen ions inside the thylakoid space that flows through our ATP synthase and synthesizes ATP. Where did the oxygen go? The oxygen will leave the leaves, and be used by organisms that consume oxygen to do cellular respiration. Also, the oxygen can actually be used in the plant to do aerobic cellular respiration as well. So they could go to the mitochondria and help be the final electron acceptor in the electron transport chain in the mitochondria for the plant. So it can oxidize its own glucose in cellular respiration using oxygen. What will ATP be used for? It's going to be used in the Calvin cycle to take our carbon dioxide and convert it into sugar. What will NADPH be used for? Same thing. It's a charged battery that can be used for the Calvin cycle. And that is light-dependent reactions. In the next video, we'll take a look at step 2, the Calvin cycle.